at the scene of crime during investigation, and also their preliminary examination at the scene of crime, because we'll be doing preliminary examination and its details a little bit, because there are times that there are misguiding evidences at the scene of crime, so you need to be sure as to whether the evidence is actually eligible to be regarded as a relevant evidence, or it is just an implanted evidence that is got no relation to the crime scene, right? So first we'll begin with a brief introduction of forensic science and what it is all about. And at any point of time, if you guys have any problem, or if you guys feel that I need to repeat anything, just let me know. Just drop a text or raise your hand and tell me at that exact point to kindly repeat anything that you want me to repeat, right? Also, is the screen visible?
Yeah, ma'am, visible.
Yes, ma'am.
Yeah, right. So, what exactly is forensic science? It is just the study and application of science to the matters of law, which means that uh, whenever any scientific principle or scientific law or any principles of natural science, be it physics, be it chemistry, be it biology, or anything, whenever such principles are applied for the purpose of assisting legal system of our country to impart justice to anyone, that principle will be regarded as forensic science. So, when the study and application of science is applied for the purpose of imparting justice, that is known as forensic science. So, it includes the business of providing timely and accurate and thorough information to all levels of decision makers in our criminal justice system. So, our criminal justice system has basically four hierarchical levels. The very first ones are the police, because they are responsible for maintaining the law and order and the peace in the society. So the very first people of that are a part of our criminal justice system is the police of any state. After that, we have the policy makers, the constitution that lays out all the roles and the rules and the laws that define what is right, what is wrong, how somebody who is not following the laws of the land is getting punished. And then we have the judiciary, the lawyers and the judiciaries. Lawyers are advocating for the interest of their clients, which can either be the person who has been wronged or the person who has wronged somebody. So they are the lawyers, and ultimately the judiciary or the judges, which are imparting justice to anybody who has been um, facing any trouble or any injustice in the society. So, are we clear about the criminal justice system levels? So, the forensic science is basically aimed at providing timely, accurate, and thorough information to all these levels of the criminal justice system. The forensic science provides evidences for the police to collect. They provide the relevant aspects of any crime scene, the indications to the police to identify and track the criminals. They take the criminals to the court. They help them, or they allow them to have an advocate or a lawyer for themselves who is going to study their case and present all those evidence and facts in front of the judiciary. And the judiciary is going to analyze and summarize whatever evidences have been provided to them and ultimately decide whether the person is guilty or not. Right? So the forensic word has basically been derived from the Latin word forensis, which is meaning, which actually just means that anything or an area which is a public place where the Roman judges or senators used to sit, debate, and hold judicial proceedings, right? So, forensic science is basically also known as criminalistics, but we should not confuse the term criminalistics with the term criminology. So, the criminalistics or forensic science is the scientific examination of any physical evidence for the purpose of helping law to impart justice. But criminology deals with the psychological angle of crime. It is basically the motive behind a criminal's act, the traits that define him as a criminal, and the behavior which indicates that he has a criminal tendency or he has a tendency of committing repeated offense. I hope I am clear to you guys, right? Matlab, it's not just that I am speaking and you guys are not understanding anything. I guess you're open to messaging, right? You have the option to message. Kindly let me know whether you're getting what I'm trying to um, convey here. I'm in the messaging box, or if you have the permission to speak within the lecture, you guys can also tell me through there. Otherwise, am I clear this far?
Yes, ma'am.
Right. So basically, criminal and criminalistics is what basic difference is. Criminology is all about psychology, and criminalistics is the scientific approach to crime scene. So we work on a few basic principles of forensic science, and the most important and the number one principle that on which the whole forensic science is based is known as the principle of exchange, or we also call it as the Locard's principle of exchange. You can see here I'm highlighting, this is the name of the scientist who gave this principle. His name is Edmund Locard, and he stated that every contact leaves a trace. This is the law. Every contact leaves a trace, which means that whenever two objects are coming in contact with each other, there is an exchange of their matter. Like, even if it is in the nano level or micro level, it happens. If you are holding somebody's hands, his or her sweat is going to be transferred to your hands, and your sweat is going to be transferred to his hands. If you're walking on a muddy road, the mud is going to have your shoe prints or footprints, as you can see here, and your foot is going to catch some of the mud that you're walking on. So if you are living by the sea and you're going out without telling your parents, and you're going out to the sea to have some fun after bunking your college, and you come back home with a lot of sand on your shoes, your mother can tell that you have been near the sea. There is no way you're going to hide that. So whenever you are coming in contact with anything, irrespective of the nature, you are going to leave your traces behind, and that object is going to leave a trace on you, which is going to prove that you have been present at a certain place at a certain time, or you have been in contact with a certain object at a certain point of time. And this is exactly how we catch criminals, be it fingerprints, be it hair, be it saliva, be it sweat, be it blood, footprints, fingerprints, whatever uh, evidence that we find at the crime scene is based on this principle only, that whenever a criminal is coming into a crime scene, he definitely has to leave somewhere there, something over there, something that belongs to him, and something that is going to help the police track him down, right? So the second principle is the principle of analysis. So the principle of analysis basically says that you will get the result of something that you have collected. If you are collecting uh, a stale fruit, and you're going to eat that fruit, You're going to have an upset stomach, and it is not going to taste good. So if you are collecting a poor quality of sample from the scene of crime, you cannot expect the analysis of that sample to be something that you wish to have. If you want to catch a criminal and get him convicted, you need to be sure that you are collecting evidences that are bang on point and that would give you the results that you're expecting. You cannot expect contaminated evidences to give you accurate results. You cannot expect degraded samples to give you the quality of result that you want. So you should be very careful in collecting any sample that is there at the scene of crime. The next principle is the principle of progressive change, which basically means that anything, whether it is man made or natural, they undergo a certain change with a change of time. So, if you have purchased, say, suppose a new phone, and you are very particular about that phone for a month or two, you're very careful that you're having a screen guard on it, you have a cover on it, and you make sure that it does not get scratched anywhere or it doesn't fall off your hand. But with the period of time when you get used to that possession, ultimately you become very casual, and the look of that phone, it starts getting old. So the look of your phone 6 months after you have purchased it is very different from what it was when you just opened it out of purchase. So no matter what, be it a chair, be it a table, a tube light, or anything that you possess, anything that is naturally present in the nature, or something that the humans are making, everything is going to undergo a slow change with the passage of time, and it is inevitable, right? Even your handwriting changes with time. Two of your signatures cannot match exactly. And if there is 100% similarity between two signatures of yours, there is almost 99% possibility that somebody has traced your signature, and it is a forged signature. It is not your original signature, because no matter how hard you try, you cannot have an 100% similar signature. Leaf signature, you cannot make an alphabet similar if even if you write it for 2 to 3 times. There has to be a deviation, right? So that progressive change is the natural law of nature. You cannot stop anybody from degrading or changing its appearance. So everything is going to change with time. Law of individuality states that anything which is natural or man made has its own individuality. No two things are identical. The same thing that I stated before. No two fingerprints are identical. No two signatures are identical. No two footprints are identical. No two pages of your handwriting are identical. Even the identical twins, except for the identical twins, uh, only the DNA of these twins are identical. Otherwise, even if you look into the fingerprints, even identical twins do not have the same fingerprints. There is some scope of separating them on the basis of the fingerprints. So, fingerprint is an absolute science. No matter how close you are related to somebody, you cannot have the same fingerprint on the same finger of the same person matching with anyone else in the world. There has to be one difference or the other. So that makes everything in this world very uniquely and particularly individual. Something cannot be duplicated, and if it is 100% duplicated, it is bound to be forged, bound to be not real, bound to be made by somebody who's trying to impersonate you. Am I clear or am I going too fast? Are you guys able to catch up to what I'm trying to explain here?
Yes, ma'am.
Great. So, the next principle is the law of probability. So, it states that out of all the possibilities that are there about commission of a crime, the chances of occurrence of a particular event is exclusive. So, suppose there is a crime and There's a suicide case. On the face value, it appears to be a suicide case. Room is closed from the inside, and a girl or a boy is found hanging from the ceiling or the ceiling fan with a rope in his neck, and he's dead. So the possibilities are two. One possibility is that he actually committed suicide, and the next possibility is somebody killed him and hung him uh, from the fan, and then they went away. But we say that the room is locked from the inside. So what, according to you, would should be the case in such scenario? What would you assess? Is it a suicide or is it a murder? Any one of you? Any one of you? If you are not comfortable in speaking out, you can write it in a text. So
Madam,
the
can you please repeat?
probability. Sorry, what?
Can you please repeat, madam?
Repeat. Okay. So I have entered a crime scene, right? The crime scene is there. The room is closed. The room is latched from the inside. There is a girl who is hanging from the fan and she's dead. So, what do you think? This is a suicide or a murder? She died herself, or somebody killed her and hung her from the ceiling fan?
There is both of the chances, no, madam.
How are both of these correct?
Maybe someone came backside of the room.
Everything is closed. There is no possibility of anybody coming inside.
Then it will become suicide, madam.
Are you very sure?
If everything is closed, and then no one will come, no, ma'am, inside.
Okay. So majority chances are of suicide, okay. But you need to look very closely. So on in investigating. So basically, according to the law of probability, 50% chances is of suicide and 50% chances is for murder. Right? Half possibility. Are you getting my point? The probability of an occurrence of an event is relative to the total probabilities of all the ways in which an event can occur. Okay, so there are two possibilities here. Either it's a suicide or it's a murder. There is no other possibility. There is no other option of asphyxiation or there is no other option of drowning, right? Either suicide or murder. This asphyxiation and drowning, these are all subparts of murder. ठीक है. So I'm just going off the track and giving you a very small example that I encountered in the scene of crime. So there was a room, and in the room the girl was hanging from the fan here, and the door, it was locked from the inside. So initially, like you all guys thought, everybody thought that okay, she has committed suicide. But on very close investigation, it was found that the doorknob here, the bolt-like doorknob or the latch on the door, they had certain brown-colored hair-like structure. Brown-colored hair-like something attached. So when they examined it under the microscope, they found that this is horse hair, horse pony hair, horse tail hair. So the horse tail hair is one of the strongest natural fiber. So this case was ultimately decided to be a murder where the people had murdered the girl, hung her from the fan, tied a horse hair to the latch, closed the door, and pushed the hair from here so that this automatically got closed, giving an impression that the girl has closed the door from the inside, but ultimately they had made such a pulley system that while pulling the horsehair, the door got locked and they were saved of the murder. So this was after around a year and a half. that the investigation came to this conclusion that this is not a suicide and a murder. But again, matlab, basically, I'm just talking to you about probability. I remembered an interesting case that I had encountered. So I thought I should share it with you. So, on the face value, never assume anything. In forensic, you're not supposed to assume anything. You should go with a doubt in your mind. You should always question everything, not believe in hearsay, and minutely examine whatever evidence you are finding. Right? Am I clear? Is it clear?
Yes, ma'am.
Right. And the last one is the law of comparison. So, law of comparison states that only likes can be compared. I can only compare a girl with a girl. I cannot compare a girl with a boy, or I cannot compare a left foot footwear with a right foot footwear. If I have to draw a comparison, I need to get a left foot footwear of that particular criminal and match it with his left foot footwear only. Left and right do not match. Left hand and right hand have no comparison. Animal hair and human hair have no comparison. If you need to compare anything, you should find the similar comparator. Uh, Otherwise, in the court of law, they're going to ask you, how can you compare two things which are not similar? You know, you should not compare anything which are not similar. Only likes, only two things that are common are the ones that can be compared with each other, right? Uh, we'll go into the different quickly look into the different units of forensic science. We have physical science unit, which is dealing with chemistry, physics, and geology for the identification and comparison of crime scene. Biology unit, which is dealing with all biological samples, which include blood, saliva, sweat, urine, semen. So these examination of all these, including the DNA examination of all these, are carried out in the biology unit of forensic lab. Ballistic unit is used for examining firearms, discharge bullets, cartridge cases, shotgun shells, and ammunitions of all types. Document division is used for paper or any other security document, which includes passports, visa, credit cards, currency notes, mint coins, anything and everything which is dealing with any security document or a document which has a handwriting, a signature, or any message written on it is going into the document division. We have the photography unit, which is dealing with the crime scene photography and evidence photography. Toxicology division, which deals with the toxins found in the body of anybody who's dead, to identify whether any external substance was administered to cause the death of that person. Fingerprint unit is there, polygraph unit is there to detect if somebody is lying or not. Voice analysis unit is there, where nowadays deep fake and AI are creating voices of people who have actually not spoken anything on that sample. So we do the voice print analysis to identify whether a voice sample is actually belonging to a particular individual or not. We also have an evidence collection unit, which is known as the scene of crime unit nowadays, that are working with the police of every state under section Bharatiya Nyay Nagrik Suraksha Sanhita 176(3), which mandates compulsory presence of crime scene forensic scientist at any crime scene of an offense which is punishable for 7 years and above. I hope you are aware of it. The section 176 subsection 3 of BNSS mandates that every crime scene of a crime which holds a punishment of 7 years and above should mandatorily have a forensic scientist at the scene of crime for the purpose of collection of forensic and scientific evidence. Am I clear? Am I clear, or was I audible?
Yes, ma'am.
Right. Right. So we have forensic entomology nowadays. Anthropology is there where we are dealing with the bones, identification of the gender of the bones, ethnicity of the bones, identification of the race, identification of the age of the person to whom these bones belong. And forensic entomology is basically study of all the flies or all the microbes that are growing on a dead body in order to estimate the time since death, because there is a successional life cycle of all the insects and other microorganisms that are growing and feeding on the dead body, and they appear over there after a particular amount of time. Each species has its own significance and its own time when they appear on a dead body. So, studying them can also help us understand the time before which the person has died. Forensic psychiatry, as I told you, deals with the psychological aspect of the crime, the mental health of the criminal, the cause of the criminal behavior, the factors that contributed to turn him into a criminal, as repeat offenders and analysis. How can we rehabilitate them? How can we rehabilitate and return them to the society? And their corrective and punitive measures that can be taken. Forensic odontology deals with the dental study of an individual, which includes bite marks and DNA examination from the tooth. Also helps in the age estimation of evidences that are found. These are anthropology and odontology are more helpful in the cases where there are mass disasters, when mass people have joined and they died, and mass people need to be identified, and the DNA needs to be understood to understand who is the person who's dead, right? Forensic engineering is also an upcoming field where you analyze the construction accidents and the cause and the origins of explosions and fires in any civil setup, right? So, basic of forensic science is done here. Do you guys have any other query in this part? If not, we'll move on to the next. Any doubts? Uh, no doubts? Great. Great. Okay. So we will move on into the next section. I'll give you a brief biological. Is my screen visible to you all?
Yes, ma'am.
Great. So, what is a crime scene basically? A crime scene is any location that may be associated with the committed crime, or where the crime has been committed. When the crime is committed, that place itself where the dead body or the occurrence has happened is regarded as a crime scene or a primary crime scene. And if there is an evidence that has been thrown away at a distance from this primary crime scene, that area where relevant and additional evidences are found is also regarded as a crime scene, also referred to as the secondary crime scene. So, primary crime scene is the location where the crime has actually occurred, and secondary crime scene can be any place or vehicle or any area where any additional evidence is hidden, or where any other relevant evidence is recovered, which is ultimately helping us in the process of investigation. Right? So, plays a very important role. Evidence is very important it is helpful in linking the criminals with the crime and ultimately helping in their conviction. So there are four types of evidences. Physical evidence is the combination of trace elements, blood spatters, visual understanding, voice recording, criminal records, anything it is that is tangible, that can be felt or seen, is falling under the category of physical evidence. Biological evidence is anything which comes from a living organism. Botanical evidences or pollens are also biological evidences. Animal hairs are also biological evidences. Human sources like blood, hair, semen, saliva, urine, sweat, everything is falling under biological evidences. Even bones and tissues and teeth are also biological evidences. Now, circumstantial evidence is the evidence that relies on an inference to connect it to the conclusion. So basically, fingerprint is regarded as a circumstantial evidence because fingerprint in itself is not giving you an indication. But when you analyze the fingerprint, when you compare the fingerprint with your database and you find a match of that fingerprint in your database, then it is helping you infer or draw to a conclusion that okay, this fingerprint belongs to this repeat offender whose fingerprint is already in our database, so this crime has also been committed by that person, right? So anything which directly is not an evidence but it relies on the inference or an or a conclusion to make it relevant to any crime scene is known as a circumstantial evidence. And invisible evidence is referred to the evidence at a crime scene that are not visible to the naked eye. Like we can come across crime scenes where blood was spattered and somebody washed the blood off. Or we can come to a crime scene where footprints are there, but the footprints is not visible. It is like the fingerprints with the sweat. So you need to have special visualizing instruments to look into the crime scene while approaching as a forensic scientist and crime scene expert, and then locate such invisible evidences and collect them for the future investigation. Right? The most important biological evidence is blood. It is a very special connective tissues, and in vertebrates, it is composed of blood cells suspended in blood plasma. It sends antibodies and fight infection and contains clotting factors and other stuff that is there in blood. So we have platelets, we have RBCs, which is having hemoglobin and it is carrying oxygen to the body. And we have the white blood cells that fights the infection. These are also the leukocytes. So the very interesting aspect of blood and its examination and its further DNA examination is the RBCs are non-nucleated. There is no nucleus in the RBC. And as you know, the DNA is present in the nucleus. So at any point of time, do not think that liquid blood is there and it is the best source of DNA. The red part, the hemoglobin part, is not giving you any DNA. It is the WBCs that are giving you the DNA that we are analyzing in the lab. The WBC nucleus is the source of DNA in any blood at crime scene or human body. So it is not the red blood cells that gives you the DNA, it is basically the WBC, right? So, analysis of different aspects of bloodstains can contribute to clarify the circumstances under which some violent crimes have been committed. So, like if you find a wall which is having a pattern of blood, indicating that a very sharp knife was used to suddenly stab somebody with a very pointed part of the knife, and the blood gushed out of that person's place of impact and it spattered on the wall, you will be able to identify the weapon of offense, the height of the offender, at what was the approximate height of the person who has hit the person with the knife. Because according to the height, the spatter is going to begin at almost that point only, or somewhere up and down. You'll get an approximate idea as to what could be the height of the offender. You could also get the idea about the posture, the location, the angle at which the person who has died was standing. So you get an idea, get a lot of idea by studying these patterns at the crime scene. You can identify the individual who has committed the crime by analyzing the DNA, and you can also identify the blood group and other basic aspects. Whether at all the liquid found at the crime scene is blood or not can also be identified by preliminary testing, because there are a lot of times where the person tries to falsely implicate someone by pouring ketchup or maybe an animal's blood or maybe any pink dye, claiming that this is blood. So, first, you need to separate out that. You need to identify whether the stain present at the crime scene is actually blood or not. And then you need to identify whether that blood belongs to the particular individual who has been implicated or not. And then after that, you need to specify all the details that are there. Right? Am I clear?
Yes, ma'am.
Great. So, what are the primary tests of blood? I'm talking about the preliminary examinations that we conduct at the scene of crime before collecting any evidence. So, the physical examination needs to be done with the UV light at 230 to 265 nanometer of length. Um, the presumptive test or the preliminary tests are done to as they produce a color reaction of hemoglobin with a certain chemical to give us an idea that okay, iron and heme is present in this liquid and this could be blood. After preliminary test, we never write in our crime scene report that this is blood. The confirmation of blood is also obviously done at the forensic lab after confirmatory tests. Preliminary test is just to get an idea that okay, this is not paint. It is giving a positive test for this result. That means it could be blood. So we need to send it for further examination to the FSL. So we need to collect this as an relevant evidence. So the first important test that we perform is the phenolphthalein test or the Castlemeyer test. So we're working with the two solutions. First is the stock solution that we prepare of phenolphthalein, which is 2 gram, potassium hydroxide 20 gram, distilled water 100 ml, and zinc dust is 20 gram. The working solution with which we use for at the crime scene for the purpose of detection of blood is ethanol, phenolphthalein stock, and 3% hydrogen peroxide. How do we perform the test? We take a small cutting of the swab. You can see the swab here. This is a cotton swab, so this would do. We'll take a small cutting of the swab or extract the suspected blood stain and place it on a filter paper. This is a filter paper. 2 or 3 drops of ethanol are put on this swab. And after that, the working solution of phenolphthalein is added and 3% of hydrogen peroxide is added. If there is an intense pink color like this, then it means that the evidence or the sample that you have found at the crime scene is can be blood. There can be false positive tests also. We cannot confirmly say that this is blood. We say that it can be blood. Right? Am I clear? Have you understood the test procedure here?
Yes, ma'am.
Okay. So this is how we do it. This is the red stain that we find at the crime scene. We take a cotton swab. We add ethanol and phenolphthalein and then hydrogen peroxide. If it stays the same as blood, it is not blood. If it turns pink, it is blood. What is the working chemistry here? The Castlemeyer test basically relies on the iron in the hemoglobin. The iron in the hemoglobin promotes the oxidation of phenolphthalein um, by the release of nascent oxygen from hydrogen peroxide. So H2O2 is a very unstable compound, and whenever there is an oxidation reaction, it readily releases nascent or unimolecular, not O2 is a bimolecular existing in a bimolecular state. So it when we have a unimolecular state of oxygen, it is known as nascent oxygen. So whenever there is nascent oxygen release, the reagent gets oxidated. And whenever there is an oxidation, the phenolphthalein is an indicator. It is a pH indicator. It will turn pink in the presence of oxygen. The phenolphthalein initially is colorless, but when it is having an acidic or oxygen rich environment, it turns pink, indicating that it is blood. So we have
That,
tetramethyl benzodic. Yes.
so the iron test may also give a false positive result, right?
Yes, that is why we are calling it as a preliminary examination. We are not saying that it is a confirmatory, because we can get iron positive in other aspects too. If there is soil and the soil has blood, if you test the soil individually, it will also give you positive result for phenolphthalein, because it can have iron traces, right? So that is why I'm saying that these are just preliminary separators. Preliminary tests are performed only to identify whether an evidence should be collected for further procedure or not. Clear?
Okay, ma'am.
So we have tetramethylbenzidine, TMB solution. Initially, we used to have benzidine, but benzidine is cancer causing in nature. So now benzidine is banned all over the country. We use tetramethylbenzidine, and we have working solution of TMB and acetate buffer. So we take 1.5 gram of benzidine, 13 gram of glacial acetic acid, and 57 ml of distilled water. The benzidine solution is ready. So we place the cutting or the swab on the filter paper, and we drop TMB solution on it and 3% hydrogen peroxide. And if we have this immediate bluish green color, then this may indicate that blood is there in that particular sample. So I'm giving you this PPT. I'm showing you this PPT so that you can again get an idea, the practical idea of how the test result looks like. So this is because the peroxidase activity of hemoglobin again. Hemoglobin in the blood exhibits peroxidase activity. Peroxidase activity means release of nascent oxygen from hydrogen peroxide, reducing it to water and creating a highly oxidizing agent, that is nascent oxygen, which can ultimately turn any environment acidic, and this acidic environment can give a color test which can indicate that the blood may be present in that sample. This is what a crime scene kit looks like. This is the TMB testing kit which the crime scene experts carry. In cases where the blood has been washed off and you cannot see blood with naked eye, you use luminol at the scene of crime. So luminol is nothing but sodium perborate, 3-amide diphthol hydrazide, and sodium bicarbonate. Yes, dear plants and some variety of fruits, vegetable oils, a lot of give a lot of components and compounds give positive for this test. That is why this is not confirming blood. This is just presumptive or preliminary separation. Preliminary screening, as we call it. We are just screening the evidence for the next procedure. The reagent are formed by taking 0.7 gram of sodium perborate and 0.1 gram of 3-amino hydride, and is mixed with 5.5 gram of sodium bicarbonate. So this is the luminol reagent. What we do, we take a suspected stain and add a few amount of luminol reagent. Appearance of fluorescent color indicate positive test. You completely dark that area where you feel that the blood might be present. You spray the luminol reagent on it, and you will see fluorescence like this. This fluorescence that is happening. This is fluorescence. So you'll see the fluorescence. This is the luminol kit head that is there in the crime scene. This is what we have. And this is used for areas where the blood has been washed off and the blood is not visible. And We do have identification of blood now, dear. We do have immunological studies, but they are kits, and kits prepared by any company which is not disclosing its component is not too admissible at the scene of crime, because we have hexagonal OBTI. There's something called as hexagonal OBTI. Okay, we have a kit now. This is hexagonal OBTI. So this kit is just not, like a pregnancy kit. There is a well here and there is an indicator here. So we wet the blood stain, we put it in the well, and then two lines appear. If these two lines are appearing, that means this is blood. Then we can send it to the FSL. But this kit, what is this reagent? What is the composition of this reagent? What is the nature of the filter paper here? What are the antibodies that are present that are giving this test positive? These are not disclosed by the company. So anytime defense lawyer can say that how is this possible? Why is this possible? Why are the other law labs not doing this? So um, in order to not create such problems, we follow the traditional and standard methods. So that is why we don't do instrumentation and kit aspects.
Okay, ma'am. But actually, I studied somewhere else that there are some advanced techniques like
There are, but they are not in India.
not in India. They are not
Yeah.
even implemented now, but may in future.
FBI, Scotland Yard, foreign, agencies are using it, but India is a bit behind. We'll come there. We'll come there slowly, but then, the lawyers. So you have to be aware, and you have to think about the proceedings and about the other person not getting the criminal not running free. So because we need to think about that, we follow traditional protocols that have been laid out by DFS, because we have a DFS manual. Nobody can challenge that manual.
Yes, ma'am. But we can change those protocols and procedures, na, ma'am, for
That is a
more
long
accurate.
way to go. We have the chief there, we have DFS, we have NFSU. There are a lot of people who are there who need to make those changes, and maybe in the next 10 years. We have DFS, NFSU, and a lot of other agencies involved, and they are working on it. Maybe in the next 10 years we'll be there, but for now, that's what it is. Maybe you guys can come up and make those representations. Meghna, you are not there in the scene of crime. You know the kind of implantations of evidence that is there. It gets very difficult to identify the real evidence that is actually connecting the crime to the criminal. So it is very important to screen the evidences. Otherwise, we'll be sending 100 articles to the FSL. We need to send only the relevant articles, and to identify the relevant articles, the preliminary screening is there, and it is just a 2-minute procedure. It is not wastage of time. It just helps you identify the course that you need to take further, because ultimately now it's all about DNA. Whatever you need, you perform the DNA examination. But if it is a paint or a ketchup at the crime scene and you just do not test it and send it to the FSL, and there is no point of performing that costly DNA examination also. So it is very important to screen your evidences and identify that you are sending relevant evidences only, and not wasting time of FSL, because that instrumentation and kits are very costly. So, anybody else has any query? We are done for the session today. We'll carry this on in the next session. Anybody has any questions, they can unmute themselves and ask. Although we've had a lot of questions in the messages, but if anybody has any other doubts or queries or suggestions or advice, they can ask.
Ma'am, it's somewhat related to our topic only, but why we are not having those publicly accessible manuals for those micro traces like falling grains? diatoms and starch grains?
Because those protocols are basically in the they're available in the botanical studies. For forensic studies, do not require a specific. I mean, you don't need a specific guideline for preparing a permanent slide of a pollen, do you? You don't. You know that a xylene slide needs to be prepared. Anybody who is well versed in botany or zoology, they know that a permanent slide is prepared in such a way. You don't need a forensic manual for that. Also, you know how a pollen is collected, of how a pollen should be collected. This is all there in the pure sciences manual. We need manuals for something of the protocols of which are not available for the pure sciences aspect. You don't have a manual for calculating the range of fire, do you? We studied that in physics when you are in 11th and 12th. You don't need a specific manual for that. Although we have a velocity and range calculator machine which is working on the principles of physics, but that's not there in the manual.
Ma'am, but for analyzing these micro traces, we need a manual, na ma'am. Like, based on that manual, based on those protocols, we have to go, na ma'am. If
But it's
like
there. DFS has the manual. I don't know why you are. I mean, we have all the manuals with us. DFS had it in the download section. I don't know currently, but DFS has never not
sorry, ma'am.
shared.
Actually, from the past few months, I'm just working on that only, but I'm not getting that. They are
Then
not
you need
public.
to get in touch with a personnel at DFS or send them a mail. They'll send you. They do not have such secrecy regarding the protocols and manuals. Or you can you are from which state?
Andhra Pradesh.
You can connect with your state lab and they'll provide you with the manuals.
Okay, ma'am. I will try that.
You can just send them a mail and they'll say it.
Ma'am, the DFSS manual we can download directly from the website,
Yeah,
ma'am.
that is what I'm saying. The DFSS manual
They
is
are
there.
not in the website, official website. That's why I mentioned that there are no publicly accessible manuals for these micro traces, like diatoms, pollen grains, and starch grains.
Let me see if I do have the manual. I'll share the datum aspect with you. I do have that. If I find it, I'll share it with you.
Like for those analysis, I'm just basing on
You
the
connect
scholarly
with your
articles.
you connect with your local state lab.
Uh, but
You'll
we
get the
can't
idea.
just rely on those scholarly articles, na ma'am.
No, don't rely on articles. Just go to your state lab and ask them. They'll give you. They'll be happy to help.
Okay, ma'am.
All right then. Thank you.
Thank you, ma'am. Ma'am, can you please drop your number or your email address?
You can connect with your mediator and they'll share the details.
Sure, ma'am. Thank you.
Thank you, ma'am.